Good afternoon, and welcome to the Center Stage Seminar Series events. My name is Tiffany Willoughby, Education Programs and Outreach Manager in the Office of Research at the University of Texas at Dallas. I will be your host. Joining me today is Dr. Theodore Ted Price. Dr. Price is the Eugene McDermott Professor and Director of the Systems Neuroscience Program in the School of Behavioral and Brain Sciences at the University of Texas at Dallas. He is also the Director of the Center for Advanced Pain Studies at UTD. He completed his PhD work with Chris Flores and Ken Hargreaves at the UT Health San Antonio, and a postdoctoral fellowship at McGill University with Fernando Cervero. He started his independent laboratory in 2007 at the University of Arizona School of Medicine and moved to UTD in 2014. His lab is interested in molecular mechanisms driving the transition to chronic pain with a focus on drug development for chronic pain disease modification and on peripheral and central mechanisms of neuronal plasticity in response to injury. He has won numerous awards, including the John C. Liebeskind Early Career Scholar Award from the American Pain Society and the Patrick D. Wall Young Investigator Award from the International Association for the Study of Pain. Ted serves on editorial boards for leading pain and neuroscience journals such as Pain and Journal of Neuroscience, and is a standing member of the Somatosensory and Pain Study section for the National Institutes of Health. Please join me in welcoming Dr. Price as he takes center stage in the spotlight. Dr. Price, welcome.
Thanks very much, Tiffany. Thanks for that really nice introduction. I'm going to share my slides. Okay, so thanks everybody for joining us today. I'm going to take you through some of the work that we do in our laboratories in BBS and in the Center for Advanced Pain Studies. I'm going to tell a couple different stories about the ongoing work in our lab and about how we're trying to come up with better ways to treat pain. So first, I have a couple of disclosures. I'm, in addition to a professor at University of Texas at Dallas, also involved in entrepreneurial activities. And I'll talk a little bit about those today. I actually think this is a really important part of what we do in research in this field. One of our goals is to take the things that we discover in the lab and turn them into products that actually help patients. And myself, along with Greg Dusor, have been involved in starting several companies. Today, I will actually talk a little bit about 4E Therapeutics, which is one of the most recent ones that we started. So this is a picture of not the entire Center for Advanced Pain Studies, but three of our labs together. Greg Dusor, who I've collaborated with for more than 20 years now. Greg and I were graduate students together with Chris Flores and Ken Hargreaves at UT Health San Antonio. We went different directions to do postdocs, and we've continued to work together since then. We both started at University of Arizona at the same time. We've run our lab jointly now for 13 years. Also pictured here is Dr. Michael Burton, who is a relatively new professor in BBS. He runs his lab mostly interested in neuroimmune interactions and pain. He's also very interested in pain and aging. He has other projects not related to pain, but he's a really important part of our group. And the three of us collectively, along with Zach Campbell, were the core investigators involved in the foundation of the Center for Advanced Pain Studies. And we have a number of other professors within BBS and the School of Natural Sciences and Mathematics, as well as the engineering school, that are integrally involved in CAPS. So within BBS is Dr. Sven Kroner. Within NSNM is Dr. Tae-Hoon Kim. And then in engineering and the engineering school is Zinping Chen. And then we also have an affiliate member at UT Southwestern, Dr. Helen Lai, and we have also just recruited Dr. Ben Kolber from Duquesne University to be joining us at UTD. So we're really excited about him joining us in BBS and being a new member of our Center for Advanced Pain Studies. All the work that I'll tell you about today was funded by startup funds to Greg and I when we came to UTD. A little bit from the Rita Allen Foundation, which is a foundation that supports basic pain research. And then the vast majority of our funding comes from the National Institute of Health, in particular the National Institute for Neurological Disorders and Stroke. And that particular institute of NIH has treated Greg and I very, very well over the years. And in terms of the data that I'll show you today, I'm going to talk a little bit about data that was generated by several different PhD students and postdoctoral fellows in our laboratory. I'll start with a little bit of data from Candler Page, who's a PhD student who actually has her own NIH grant. She's getting really close to graduating. I'll then show you a little bit of data that was generated by Pradipta Ray and Andy Wang Zhu. Pradipta is a postdoctoral fellow with us. Andy is a PhD student. And then towards the end of the talk, I'll talk about the work of Stephanie Shires, who was a PhD student with me, but has since completed her PhD and is staying with us doing a postdoc. So we're interested in pain, and we're really interested in understanding what causes people to experience pain at the molecular level, and how can we use that information to help us better treat pain. So this is a painting called A Stab in the Arm, and if you've ever heard me talk before, you probably know that I really like this painting a lot. I like it for a very specific reason. I think it portrays the problems that people have with pain very well, and it also does a nice job of describing at least the basics of the neuroanatomy. So if you were to imagine that this man was actually just stabbed in the arm, these would be the pain sensing neurons, which we call nociceptors, that innervate that area of his arm. And they're red because they're sending an electrical signal to the spinal cord. These peripheral neurons then enter the central nervous system at the level of the spinal cord. So they go into the spine here, and then they send a signal on up to the brain. And the signal actually disperses throughout many different areas of the brain. And receiving a pain signal in the brain is one of the most important things that can happen to you in terms of setting off the alert within your brain. So it causes an immediate reflex reaction that tells you to get away from whatever it is that's hurting you. And if you actually sustained tissue injury from what occurred to you, it also tells you to protect yourself throughout the course of that tissue injury, which is really important because if you don't protect yourself from injuries like this, they can result in many different bad things happening. For instance, you can get infection and end up losing your arm potentially if you don't protect a wound while it heals. So the pain system is really good at that. So it serves a definitive purpose, at least acutely. The problem is that that's not what happened to this guy at all. He actually had a spinal cord injury, and when he had that spinal cord injury, it injured the peripheral pain sensing neurons. He didn't have any injury in his arm, but he felt like he felt like he was being stabbed. So he had a surgery to fix his injured vertebrae, and the surgeon successfully did that. Unfortunately, when he woke up from the surgery, his pain was not gone. And that actually is a common occurrence in people that end up with neuropathic pain. They'll have some kind of injury that causes a lesion of their nervous system, usually the peripheral nervous system. They can have whatever caused the lesion repaired, but the lesion to the nervous system is very difficult to repair itself. And unfortunately, that causes these neurons to change their phenotype such that they become active over a very long period of time, and that drives these types of neuropathic pain. So this is a man. This is one of the most famous female artists ever to walk the earth, Frida Kahlo. So Frida Kahlo was also a pain patient. She had a very severe injury when she was a teenager, a late teenager in Mexico City. She was on a trolley, and that trolley train was hit by a car, and she had a severe spine injury. That spine injury, she had many, many surgeries. In fact, this particular painting, which is called Without Hope, was painted in reference to her experience after one of these surgeries. And you can see that she feels like her insides are being removed from her. The pain is so severe. So why would I show you the picture of the experience of a man and of a woman? Well, one of the things that I want to highlight in this talk is that while the pain experience may be similar between these two people, the underlying molecular mechanisms might actually be quite distinct. And we think that that's really important to understand in terms of developing new pain therapeutics. So one of the things that we're trying to do in our laboratories is to better understand in particular how chronic pain develops in women, with the idea that we're doing a particularly bad job of understanding the molecular mechanisms that cause chronic pain in women, and that if we had better therapeutics that could target this specifically, we could do a better job of treating chronic pain, both in men and in women. So I'll give you a little evidence behind that idea today. This is me when I was a young teenager. I'm showing you this picture because these were three of my very best friends when I was growing up right here in Dallas. This is actually in the old West End. This is before they completely built it up. There used to be trains that actually ran through there, and this was during a Hoop It Up back in the late 80s or early 90s. So Chris Nelson, who's pictured right here, we played many sports together, and he had a really bad shoulder injury when we were in high school playing football. And much like the two examples that I just gave you, Chris suffered really badly, especially as he got older, from chronic pain. And in fact, he had a surgery when he was in his late 30s to try to fix this, and the surgery didn't go well. Unfortunately, Chris became addicted to opioids after that. He got clean and was doing relatively well, but unfortunately, about 4 years ago, he overdosed and died from prescription opioids. And these two things have become incredibly intertwined: the chronic pain epidemic and the chronic opioid epidemic. And many of you probably know that we have started prescribing in the last 20 years opioids for the treatment of chronic pain, much more than was ever done previously. And this has played a huge role in this kind of expansion of opioid use and opioid addiction. And what this slide here shows is that the opioid epidemic has really grown exponentially since the late 90s and early 2000s, and that it in particular has grown in prescription opioid use. And these drugs, there's not very much evidence that they're actually any good for treating chronic pain, which is one important thing. And again, of course, they're really quite dangerous drugs that you can easily overdose from. And tragically, I lost one of my best friends to an opioid overdose. And I think that many people have been struck within their family or circle of friends by this epidemic. So I think it's important for us to recognize this particular problem in our society and do better. And one of the ways we can do better is by developing non-opioid therapeutics for the treatment of chronic pain. Chronic pain is also a huge epidemic within the country. In fact, it's orders of magnitude bigger than the opioid epidemic. It affects about a quarter to a third of the population. It costs an enormous amount of money. It's estimated at between $500 and $700 billion annually. And a lot of this ends up going to the so-called socialized part of our medical system, so to the Veterans Administration, to Medicaid, etc. So our inability to treat pain well actually impacts all of us, because we all pay for this particular medical problem in our taxes. And of course, the issue that's bigger than money is that it reduces the quality of life for the many people that suffer from it. And the truth is that we just don't have really good drugs to treat chronic pain. And I think that we can do much better here, and this is one of the major focuses for our center. People often say also, pain is awful, nobody wants to live with pain, but people don't die from pain. And that also is just not true. So some people become addicted to opioids, and some people end up dying of opioid overdoses, like my friend. But also many people that have chronic pain problems end up committing suicide. So this is particularly tragic, and it's estimated that of all the people that commit suicide every year, about 10% of them are people that have chronic pain problems. So we don't necessarily know that they committed suicide because they had chronic pain, but the strong overlap of those two is certainly suggestive. And these people usually do not die of opioid overdose. They usually die of self-inflicted gunshots. So this is really quite tragic, and I think it is probably true that pain does kill people. So moving on to more optimistic topics now of how we can actually do something about this. I'll start with something that's not very optimistic, actually, but I'll try to offer some hope around it. So we have a big problem in biomedical research on two fronts with respect to better understanding disease states in men and women. One is that the pipeline for development of female scientists is remarkably leaky. What do I mean by that? I mean that at the early stages, so like the PhD stage of training, in most scientific programs within the country, that's close to 50-50 in terms of males and females that are involved in this. But as career progression goes on, you can see that there's a major fall in the percentage of investigators that are female. So this is a big problem. So most research is led by men because of this leaky pipeline. And another big problem is that most of the studies that are done, either preclinically or clinically, have studied male subjects and not female subjects. So we only really have a really good understanding of underlying disease mechanisms in about 49.5% of the population, which is not a good thing. So NIH has done something about that over the last decade, and that's to say, if you're not going to take sex as a biological variable seriously in the experiments that you do, we're not going to fund you. So within our group, we've taken this really seriously and as a big opportunity to try to better understand underlying pain mechanisms in females. And so what we're now doing is really trying to understand how underlying molecular mechanisms might be different between men and women in the development of chronic pain. So historically in the pain field, a lot of work has been done on trying to understand whether men and women perceive pain differently. I would argue that most of that work has led to very tiny differences between the sexes that aren't very clinically meaningful. But there's much better evidence that underlying mechanisms are different, and I think that has big implications for therapeutics. So let me give you a good example here. So this is an experiment that was done by a group in Australia, and they took one of the most common painkillers, non-opioid painkillers out there, ibuprofen. So my guess would be every single one of you has heard of ibuprofen. It's a so-called non-steroidal anti-inflammatory drug. And what they did was to test this in a volunteer pain model. So they gave people a heat stimulus when they had either taken ibuprofen or not taken ibuprofen, and measured the amount of analgesia, pain relief, that they had from the ibuprofen. And what they found was pretty striking. So in males, which are shown here, ibuprofen gave a very nice analgesia, but in females, it did absolutely nothing. So striking difference in terms of a basic underlying mechanism of analgesia. And they also did pharmacokinetic experiments to measure whether the drug was actually there. You know, one possibility is that the drug was metabolized rapidly in women and not men. Some drugs that does happen, but this particular one, no, that was not the case. So there's something different about the underlying pain mechanisms between men and women that we need to better understand. So in our labs collectively, we focused a lot on neuropeptides to try to understand this, and I'll show you a little bit of data around CGRP. Which is calcitonin gene-related peptide. So the reason that we decided to focus a little bit on CGRP was mostly because of the work that Greg Dussor had been doing around this. So Greg's group studies migraine headache, and migraine headache is just far, far more common in women than it is in men. And we know from clinical studies that CGRP is increased in the blood of people that have migraines during migraine attacks. And we also know that therapeutics that target CGRP, so that remove CGRP from your blood, are effective for treating migraine headache, which again occurs preferentially in women versus men. So that led Greg's group to study whether or not CGRP might have a stronger effect in producing pain within headache areas. And they basically found that CGRP is a very strong sensitizer of neurons that cause headache pain in female rodents than it is in mice, in male rodents. So there does appear, at least within the headache area, sex-specific role for CGRP. So Candler decided that she wanted to try to test whether this was the case in non-headache areas. So what we did was to inject an inflammatory mediator into the hind paw of mice, and we used antibodies against CGRP. So these are actually antibodies that are very similar to the drugs that were just approved for the treatment of migraine. And she tested these. So this is the CGRP antibody versus controls in mice. And what she found was that acute sensitivity was almost completely absent in the mice that were treated with the CGRP antibody, and they didn't transition to a chronic pain state. So this is in females, so this is great. It suggests that we can use these CGRP antibodies to treat other types of pain beside headache. So the question is there a sexual dimorphism here? And the answer to that is there is, and it's about as striking as you can imagine. So the CGRP antibody in males does absolutely nothing. No effect on the acute phase and no effect on the chronic pain phase. So really, really striking sexual dimorphism. So what Candler's work suggests is that these CGRP based therapeutics, and there's many of them that have now been approved by the FDA for the treatment of migraine, could potentially be used to treat many different types of pain, but they might only be effective in women. So we're now really interested in trying to work with some of the companies that have developed these to do clinical trials in other types of pain disorders that primarily affect women, such as fibromyalgia. So one of the really key goals in our laboratory is to try to understand underlying mechanisms that drive pain, and then use that to develop novel therapeutics. So in this experiment here, what we're doing is we're looking at these peripheral sensory neurons that we know are involved in pain, so these so-called nociceptors, and we're recording from them in a dish. This was actually an experiment we did in collaboration with Joe Pancrazio's lab at UTD. So what we did here is to use neurons from normal mice and also mice that have a mutation in the signaling pathway that we're interested in. So first I'm going to focus on what happens in the neurons from normal mice. So you see that under normal conditions, these neurons are not active. They're not firing any action potentials. They're not generating an electrical signal. So each one of these lines is an action potential, okay, that electrical signal. So when we expose them to this inflammatory mediator, IL-6, you can see that these neurons really go kind of crazy. They start firing lots and lots of action potentials. They do this for a long period of time. And what's really interesting is when we take the IL-6 away, so this is like that surgery that I told you about that the man had after he had the spinal cord injury. They did the surgery to fix the tissue injury. Does the nervous system go back to normal? So when we take the IL-6 away, it doesn't go back to normal. The neurons continue to fire action potentials, and they sustained this increased electrical activity for a long period of time. Now, in these mice that lack the signaling pathway that we're interested in, you can see that IL-6 causes an initial response, but the transition to this new state where they're firing action potentials, generating this electrical signal all the time, is mostly absent. So this is a really good indication that this particular signaling pathway might be really important for the development of chronic pain. And targeting it with drugs might be an advantageous way to go. So this is in rodents. One thing that we've become really interested in is trying to move away from using rodents as a model and trying to work more with the human nervous system at the kind of electrophysiological and molecular level, so that we can get a better idea of if these targets we're developing can actually be employed for the treatment of pain, and if the clinical trials that we might do down the line would be successful. So this has turned into a really great collaboration with people at MD Anderson in Houston. We work with patients that have cancer that's infiltrated their vertebrae. So you can see some radiographs from a person that has cancer in their vertebrae. So the spinal cord is right here, and the vertebrae are around it. And this is the cancer. So this is a bad scenario. You don't want this to happen to you. These patients need to have a surgery called a vertebrectomy, where the cancer is taken out and the vertebrae are also taken out, and they build this structure, this metal structure, to protect their spine so that they don't have a collapse of their spinal cord, leaving them paralyzed. So this is a really unique opportunity for us because the dorsal root ganglion neurons, those peripheral neurons that sit around the spinal cord, come out during this surgery. So this gives us an opportunity to phenotype these patients beforehand, determine if they have pain or not, and then to look at how the neurons change depending on whether or not they have pain. So for instance, in some patients, like this one, they have pain on one side of their body, but not on the other side of their body. So the dorsal root ganglion that comes out from the right side, we can look at things that are associated with pain, and we have a genetically matched sample on the other side where we can see, this is our control. So one of the first experiments we've done is to try to kind of recapitulate that experiment that I just showed you that we did in mice. So here we don't expose the neurons to anything at all. We simply just put them in a dish and we see if they generate electrical activity. And remember, I told you before, if we don't expose those mouse neurons to anything, they normally don't generate any electrical activity. So you can see that this neuron is firing action potentials, it's generating electrical activity. This neuron comes from a dermatome where the person had pain. And in fact, what we find on a population level is that in dermatomes where people have pain, we see about 20% of neurons show the spontaneous activity. And from dorsal root ganglions taken from people that don't have pain, we essentially never see the spontaneous activity. So this increased excitability of these neurons seems to be really, really important for causing pain. So this is nice because it demonstrates that what we think is true from our animal models actually happens in patients that have neuropathic pain. And it also gives us an opportunity to test drugs that we have ideas that they might work from animal models directly on these human neurons, which is a really great bridge to go from a preclinical idea to a potential clinical trial. So this particular drug targets that signaling pathway that I told you about in those previous slides. It's called EFT508, and it's an inhibitor of a kinase called MINC. And what you can see is this neuron is spontaneously active. We apply the drug, and eventually the spontaneous activity of the neuron goes away. And we've now done this on many neurons from patients that have neuropathic pain. and the drug is extremely efficacious in reversing the spontaneous activity. So we think that means that it would make a really outstanding neuropathic pain drug. And in fact, that's exactly what we intend to do. So that work led to the formation of 4E Therapeutics. 4E Therapeutics is actually based in Austin. Our CEO is a guy named Craig Benson, who used to run a foundation called Beyond Batten's, which developed a drug for Batten's disease, which is a fairly rare neurological disorder. So it happens that Craig's daughter had Batten disease. So it's really amazing what he managed to do at his previous foundation, discovering underlying mechanisms of Batten's disease and then actually developing a drug. So we feel really great about being able to recruit him on for this effort for neuropathic pain. And essentially what we're doing is making modifications with a medicinal chemist that did his training at UT Austin, who's making molecules, and now we're testing those molecules, looking for inhibitory activity, optimizing the pharmacokinetics of the molecules so that we can make a better drug for the treatment of neuropathic pain. And we're now pretty far into this process and getting relatively close to thinking about choosing our clinical candidates for actually moving this into the first stage of clinical trial development, which is the investigational new drug application process. So in addition to doing these electrophysiology experiments, we can also use a technique called RNA sequencing to try to better understand the underlying molecular mechanisms that might be driving chronic pain in these particular patients. This slide looks complicated, but I'm going to ask you to just pay attention to the colors and the shapes, and I think it should become pretty clear what's going on. So things that are in yellow are genes that were upregulated in males that had pain. Things in purple were upregulated in females that had pain. The little green diamonds are genes that are expressed mostly in a type of immune cell called a macrophage. And the little red stars are genes that are mostly expressed in neurons. So hopefully now it's pretty clear that there's a lot of male increased things on this side of the equation, and there's an awful lot of these green diamonds on those genes. So we think that this is a very strong indication that macrophages play a really important role in determining neuropathic pain development in males. But there's very little signal happening in the female upregulated genes. In the females, something quite different seems to be happening. We see upregulation of a lot of neuronal genes, and many of these are receptors called G protein coupled receptors. So what we think this is really a kind of first sign that the underlying mechanisms driving neuropathic pain are quite distinct between males and females. Interestingly, the findings we have in males are very consistent with what's known in the preclinical literature. And our findings in females are actually consistent with a fairly small literature that's mostly been coming from our group, that ligands that act on G protein coupled receptors are more important for development of chronic pain in females than they are in males. So Pradipta Ray, who's really leading this effort, has done some work to try to better understand this. And basically what he's done is identified a couple of different subtypes of patients that might have different underlying mechanisms driving their neuropathic pain. So I'm just going to talk really briefly about 4 of these subgroups. One of them upregulates some neuropeptides like brain-derived neurotrophic factor and galanin. We know that these are really important for synaptic plasticity in the central nervous system. So we think that this particular subset of patients may have really strong what we call central sensitization. That's an underlying mechanism for their pain. This is interesting because there are drugs that target central sensitization. So we think that if we could identify these patients somehow, we might already have a good idea of what drugs we would want to use to improve their neuropathic pain. Another subset of patients, we see upregulation of an ion channel called TRPM8 and also a tyrosine receptor kinase called TREK-A, which is a receptor for nerve growth factor. This is interesting because TRPM8 is a cold sensor, and it would suggest that these people should have really strong cold hypersensitivity. So there might be an underlying physiological phenotype that you could find relatively easily in these patients. And there are drugs that are being made against nerve growth factor and TREC-A. So a therapeutic may be nearing approval. These are actually being reviewed by the FDA right now that might be effective in this population. So these are two populations that we might actually know how to treat right now. The question is could we identify them? So I'll get into that a little bit later, because it's unlikely we're going to be able to take the dorsal root ganglion out of your normal average everyday patient. This is another subgroup that's a little bit bigger. It has a bunch of genes that are associated with macrophages, although they're found in many different cell types. Almost all of these are males. So we think this is our macrophage driven subgroup, and that this might be a group of patients that you would want to try to target with therapeutics that would reduce this kind of macrophage reactivity. And this is another subgroup that has a different immune cell type signature, but I have to admit that we haven't yet really figured out what immune cells they are. But interestingly, this group is mostly female. So we're really actively trying to focus in here with the idea that this could give us a better idea of the underlying mechanisms driving neuropathic pain in female neuropathic pain patients. So like I just mentioned, it's going to be really hard, I think, to take the dorsal root ganglion from your average everyday pain patient. And in fact, there's no way on earth that that's ever going to happen. So we have to figure out a way to take what we're learning from these kind of rare patients that we can get access to and apply it to a broader patient population. So to that end, Stephanie Shires and Deanna Ferreira and some other people in my lab are actively working on trying to do this. So these really beautiful pictures here are taken from the sural nerve of people that are having lower leg amputations. So now we're getting nerves from lots of different types of patients. And the idea here is that we can do molecular profiling on these nerves and see if it's consistent with the changes that happen in the dorsal root ganglion, because it's easier to get access to nerves. And we can gradually move out to the point where potentially we can just sample very sparsely nerve endings in the skin, like with skin biopsies, to try to get a better idea of what's changing at the molecular level in these patients. So we're actively working on that, but we don't really have a lot of data, but that's a major direction that we're moving. We're also really trying to understand the molecular identity of neurons in the human dorsal root ganglion. So we know a lot about rodent dorsal root ganglion neurons, but we unfortunately don't know a lot about the molecular profile of human dorsal root ganglion neurons. So we're spending a lot of time trying to understand that. And I'll just give you one example of why we think this is so important. So this is a technology called RNA-scope in situ hybridization. So Stephanie here, what she's done is compare expression of a really important ion channel for pain called TRPV1 between mice and humans in the dorsal root ganglion. And what she's found here is, I think, really striking. So in mice, TRPV1 is expressed in a very specific population of neurons that we call the peptidergic nociceptor subpopulation. So a lot of what we know about pain pathways, et cetera, have used tools to manipulate this particular receptor and pathway in mice. And the assumption here is that these neurons are going to be somewhat the same in humans, but in fact, that is not the case at all. In the human dorsal root ganglion, TRPV1 is actually expressed by almost all of the nociceptors, if not all of the nociceptors. So this pathway probably plays a more important role in humans in terms of pain transduction and activation of pain pathways than it does in mice. So a really fundamentally different molecular profile that I think is important for thinking about how to better develop pain therapeutics, and also just the importance of doing this kind of molecular work in humans. All right, so I want to just wrap up quickly by talking a little bit about the work that we've done on COVID-19. So, you know, everybody's life since March has been profoundly impacted by this disease. And we thought when this first started going that we had some data sets that would really help us to maybe make some insights that could be helpful in the better understanding the clinical picture in COVID-19. So we've been doing some work on this using RNA sequencing. And it turns out that these dorsal root ganglion neurons that we've been getting from MD Anderson were really important in allowing us to do this. So but whether you know it or not, your lungs are really, really richly innervated by the nervous system. And that innervation comes from two places. One of them is through the vagus nerve. So there are some nociceptors in the vagus nerve that innervate the upper airway really densely, and also a little bit of the lower airway, although to a lesser extent in the lungs. And then your thoracic dorsal root ganglions, which are the ones that we get from MD Anderson, they also richly innervate the lungs. And of course, we know an awful lot now about the molecular phenotype of these neurons. We also know from some studies that have been done in China how immune cells within the lungs change in COVID-19. So those experiments have been done by taking what's called bronchial alveolar lavage fluid. So basically they lavage the lung with some fluid to pull some cells out, and then they've done sequencing on those. So what we've done is to compare the sequencing of the immune cells in the BALF fluid with our dorsal root ganglion to look at ligands that come from the immune cells and how they might be acting on neurons. So you could ask, why is this important? Well, what seems to be happening in people that get severe disease is that their lungs fill up with fluid. That causes this acute respiratory distress syndrome, or ARDS. And it's basically really severe pneumonia, and it can be fatal. So when you have fast pneumonia like that, it suggests that there's some neurogenic component to it. So we thought that maybe we could understand how that happens. And our results have actually been really interesting along these lines. Basically, what we found is that these immune cells in the lungs of COVID-19 patients create a lot of ligands that act on receptors, or potentially act on receptors that are expressed in the human dorsal root ganglion. And some of those ligands are CCL2, which is a chemokine, epiregulin, which is an agonist of the EGFR receptor, and some fibroblast growth factor ligands that actually modify voltage-gated sodium channels that are very specifically found on sensory neurons. So we think this can potentially give some better understanding of what's happening within the lungs in terms of neuroimmune interactions in COVID-19. It also provides some opportunities for intervention. So for instance, there are antagonists to the EGFR receptor that are used for the treatment of cancer that could potentially be used to try to block neurogenic inflammation within the lung in people that are suffering from severe COVID-19. So our idea is not to do these clinical trials, it's to just put new data out there that could potentially lead to clinical trials that could have a positive impact on disease. So another big question around the disease is whether or not the virus itself can potentially infect neurons. So I think that's really important now, because as we're seeing patients that have gone through the disease and recovered, some of them have neurological issues. And it's mysterious how the disease gets into the nervous system to begin with and how it spreads. So the receptor that the virus uses to get into cells is called the ACE2 receptor. So Stephanie in the lab has been looking at whether or not ACE2 is expressed in human DRG neurons, and the last time we made this figure it was just from one sample, but we've now done it from at least 2 samples, and the results have been consistent. So I think that this will turn out to be a consistent finding from individual to individual. So it turns out that ACE2 is expressed by sensory neurons, and it's in fact expressed by a very specific population of sensory neurons. Interestingly, this particular population doesn't really exist in rodents. It exists in primates, so it's kind of a mix of the peptidergic and non-peptidergic population. And we've now also looked at the expression of a marker for the neurons that innervate your skin. as well as the surface of your visceral organs. So it's a receptor called MRG-PRD, and it seems to overlap well with those as well. So we think that this at least suggests that one way that the virus could get into your nervous system is through your sensory neurons. And I think that will ultimately, if it turns out to be true, would have a really big impact on our ability to understand how this virus ultimately affects the nervous system. All right, so what are the take-home points from today? I hope, number one, that I've got you interested in this idea of sex differences and mechanisms driving chronic pain. I think we understand a lot better what's happening in males, but we're really interested in what's happening in females. We're particularly interested in CGRP and different types of immune cells. And we're also really trying to push the envelope as much as possible within the center on working with human tissues to try to get a better molecular profile of the peripheral nervous system. We're also hoping to eventually move into working on the central nervous system, where we really don't have very good information at all about the molecular profiling of central neurons. So we, to really facilitate this, we have some wonderful collaborations. One is with MD Anderson. We have another one with the Southwest Transplant Alliance, which is right here in Dallas, just down the street from us, actually, on Walnut Hill and 75. So I'll wrap up by just, again, highlighting the really great people that I work with that have done all the work that I told you about today. Stephanie did the RNA scope. Pradipta and Andy did most of the work on the RNA sequencing. Kendler Page did the work on CGRP. Greg and I have worked together for most of our lives now, which has been just a terrific collaboration. We're thrilled to have Michael at UTD and are super excited about the work he's doing. It's wonderful also to work with Zach Campbell and the other members of CAPS, as well as Helen Lai, who's at UT Southwestern. Our collaboration with the folks at MD Anderson has just absolutely been wonderful. There's two groups there that work with Pat Doherty and Annemieke Kevlars and Kobe Heinen. And we also have a very active collaboration with folks at McGill University and University of Arizona. And again, nothing happens at all without the support of the folks at NIH. So with that, I'll stop and take any questions.
Indeed, Dr. Price, we do have a few questions that have been submitted. The first question is, do you think neuro advancements will be more attributable to private or public funding?
So public funding from NIH dwarfs everything else. So I kind of look at a lot of the private foundation funding as being things that can help people get started when they're very, very early on in projects to get critical pieces of preliminary data. But the funding that comes from that is really not enough to do the kind of basic, very detailed work that I was talking about today. Now, once you if you find a target that you want to go after and spin it out into a company, and then really, try to develop a product that can actually make it to the market, and I really think that that's the only way to do that. That just doesn't really happen within academia, at least from the perspective of developing drugs. There you need to have a mix. So most groups end up starting again with public funding, so SBIR and STTR grants are really critical to get rolling. But if you don't raise private funds, private equity or angel investors or something like that, eventually you probably are not going to make it. So within 4E Therapeutics and within the companies that I started before that one, we've always started with getting some small funding from NIH, and then we've gradually moved towards private equity to do what we need to do to start to move the therapeutic to the clinic.
Excellent, thank you. Another question, little statement prior to, there's been some new tools for evaluating the effect of mice and rats, but our ways of inducing nociceptive stimuli don't seem to have advanced too much. I know there's been cool stuff using up to dreads, but we're stuck with the von Frey still. Any new tools in the works? And then the individual goes on to note that von Frey is an example, not to say that von Freys aren't good tools as well.
Yeah, sure. That's a great question. So there are lots of good tools. So or new tools. You can use optogenetics or dreads, obviously, but I think you're still stuck with needing to do some kind of pain measurement. So there's two things that we use that are non-evoked stimuli that we like. We use the mouse grimace scale, which was developed by Jeff Mogul. There are some automated forms of this that are available now that work pretty well. So Mark Zilka developed a program in his lab that works at least on some types of mice where you get automated grimace measures, and that works pretty well in our hands. We're kind of actively working with him to try to improve that. I feel like that's eventually going to do well for lots of people for preclinical therapeutic testing. There's also condition place preference. So condition place preference is not the best kind of experiment to do if you need to do like dose response studies or something like that. But it does work pretty well qualitatively. And I think it gives you a good indication if your drug is potentially relieving spontaneous pain. My personal view, however, is that physiology is actually still a really good way to go and quite precise. You know exactly what you're measuring. So, good old electrophysiology, especially if you can do it in mouse neurons and also in human neurons, I think is a really good proxy for behavior.
Thank you, Dr. Price. I do have an individual who is curious about your TEDS pain cream. Can you identify what makes it different than any other over-the-counter pain creams or ointments?
Yeah, so that's a pretty interesting story. So Greg and I have worked on a kinase called AMP activated protein kinase or AMP kinase for many years now as a potential target for reducing nociceptor hyperexcitability and preventing development of chronic pain. And it turns out that this particular pathway is activated very robustly by a natural product called resveratrol. So many of you have probably heard of resveratrol before because it's very famous for potentially being a life extension molecule, and I think that's definitely true in worms, also in flies, maybe even in mice. But if people take oral resveratrol, it doesn't really get out of your stomach at all. So going down to Whole Foods and buying a bunch of resveratrol pills probably does nothing for you. But resveratrol gets across skin really well, and it's metabolized in the liver. So, to avoid first pass metabolism, you can give the drug topically, and it'll work locally. So, our idea in developing the pain cream was to take resveratrol-based topical to the market and call it a pain cream by just not putting resveratrol on the active ingredients list. So resveratrol is generally regarded as safe by the FDA, and we've used methyl or trolamine salicylate as the active, so-called active ingredient in both of the products, but they contain resveratrol, and we really think the resveratrol is the key thing, and we're the only topical on the market that contains resveratrol. We contain a particular type of resveratrol, which is very important, and that's a trade secret. And we also keep our formula as a trade secret. So that I think gives us an advantage once we're in the marketplace.
Thank you, Dr. Price. We have two more questions in the queue. The first one is as follows. Pain has some interesting associations with immune cell interactions. Are there any already existing immunotherapies being tested for chronic pain treatment?
Yeah, that's a wonderful question. I don't think there's any that are out there that are being tested from the perspective of, you know, the kind of classic immunotherapies, which would be like the ones that the Nobel Prize was awarded for last year or 2 years ago. I don't remember which year it was, that are used for cancer. But there are lots of therapeutics that target molecules that come from the immune system that are used to treat chronic pain. So the best example is rheumatoid arthritis. So the anti-TNF therapies, those are disease-modifying drugs, and in many people that take them, their joint inflammation and pain goes away. And we know TNF acts directly on nociceptors to cause pain. So that's one example. Other examples are IL-6 therapeutics that are also used to treat rheumatoid arthritis, but those are being tried in some other areas now. And then the nerve growth factor therapeutics are you could also put them into this category, and those are being reviewed by the FDA right now for potential osteoarthritis and maybe neuropathic pain treatment. Finally, you know, the just classic NSAIDs are really kind of immunotherapies because they target cyclooxygenase expression in immune cells that causes prostaglandin creation. So there's lots of precedent for this. I just think that what we need to emphasize more moving ahead is the really interesting differences in the underlying biology that are sex dependent, and we need to exploit that to a greater extent.
Excellent. It looks like we have two final questions left in the queue before we wrap up today. There's tons of universities that have collaboration between medical campuses and university campuses, but this is one of the first times I've seen pretty extensive reverse translation, human to mice, and tissue availability. Is this a kind of paradigm change in basic science? What might slow adopting this pipeline down?
I hope it's a change in the way we do things, because I think we should. The technologies that have been developed that kind of enable what we're doing are widely available, and it took some real pioneers in this area. Rob Giroux, Michael Gold, some of the folks at Anabios who really kind of pushed this thing of using dorsal root ganglion from organ donors. But I think, you know, their example is out there now for people to follow, which is exactly what we've done. And that there are organ transplant procurement agencies all over the United States that you can work with to get access to this tissue. So I think the pain field can set a really good example for many areas of neuroscience, where we can try to take better advantage of the technologies that are available to change the way we do basic science, and hopefully start learning far more about the molecular specificities of the human nervous system, and how it differs from some of these other species that we've studied for so long.
Thank you, Dr. Price. So final question, if TRPV1 distributes different from human and mice, how do you use mice to develop drugs using in humans?
Great question. I think relying solely on mice to develop therapeutics to treat pain in people was probably not a good idea to begin with. And I don't want to leave you as we sign off of this thing with the impression that doing research in mice in the pain field is not useful. I think it is extraordinarily useful. We gain tons of insight into basic neuroscience that we just couldn't get any other way. But the concept that we're going to use these models to develop therapeutics, I don't think is a good one. I don't think that they're really capable of the molecular precision you need to identify targets. For that, we really need to be relying on patient tissues, and that there are these subtle or even not so subtle differences between species that I think could lead us quite astray from the idea of testing therapeutics.
Thank you, Dr. Price. We sure do appreciate your time and all of the excellent information you shared with us related to the Center for Advanced Pain Studies and your research.